Today we'll talk about the post-Mendelian genetics or neo-Mendelism. This post-Mendelian genetics, it involves the genetics which was discovered after the Mendel, what is called as neo-Mendelism. One of the important aspects of the this neo-Mendelism or post-Mendelian genetics is gene interaction. Gene interaction. Gene interaction is simply the influence of alleles and non-alleles on the phenotypic expression of an individual. So gene interaction is the influence of alleles and non-alleles on the phenotype of an individual. On the phenotype of an individual. This gene interaction can be it can be of two types. What is called as intragenic interaction. It can be, gene interaction is of two types. It can be intragenic interaction, that means also called as allelic interaction, and it can be intergenic interaction. This intergenic interaction, that means interaction between the genes, it is non-allelic interaction. So this intragenic interaction is of three types. Intragenic interaction, that means this intragenic interaction, it involves alleles, that means only one gene involved. Only one gene involved in case of intragenic interaction. It can be of three types. It can be incomplete dominance, intragenic interaction, that means only one gene is involved. It is an allelic interaction, only one gene is involved. So incomplete dominance, codominance, and multiple alleles. These are examples of intragenic interaction. So we have second one, that is intergenic interaction. This involves two or more genes. This is what is called as non-allelic interaction. This non-allelic interaction can be it involves complementary genes. It involves supplementary genes. It involves epistasis and also hypostasis. It involves duplicate genes. It involves lethal genes. It involves inhibitory genes. So these are some of the instances of intergenic interaction. So we can say that this intragenic interaction. So what is gene interaction? Gene interaction is simply the influence of alleles and non-alleles on the phenotypic expression of an individual. This gene interaction can be of two types. One is intragenic interaction, that is between intragenic interaction, that is allelic interaction between the alleles, and then we have intergenic, that means between the genes, also called as non-allelic interaction. This intragenic interaction, it involves only one gene. So we have examples of this intragenic interaction, that is we have incomplete dominance, we have codominance and multiple alleles. Then in case of this intragenic interaction, it would be like this. So intragenic interaction can be explained like this. It involves this is homologous chromosome. This is homologous chromosome. Intragenic interaction involves, for example, this is one allele, this is another allele. The interaction between these two alleles of the same gene, this is intragenic interaction. Also, it can be like this. These are homologous chromosomes. An interaction between these two genes, for example, small a. This is also an intragenic interaction, that is interaction between the two alleles of the same gene. This is intragenic interaction. Or we can have this, that is two alleles of a gene occupying the same locus on a homologous chromosome interact to produce the phenotype in case of intragenic interaction. So it would be like this: two alleles of a gene located on the homologous chromosome interact to produce the phenotype of an individual in case of intragenic interaction. That means only one gene is involved. This is intragenic interaction chromosome. Whereas in case of intergenic interaction, two or more genes are involved. It would be like this. For example, we have in case of intergenic interaction, two or more than two genes present on the homologous chromosome. This is homologous chromosome. They interact to produce the phenotype. For example, this is one gene. There are two alleles of the same gene. This is another gene. So these two genes interact. These two different genes interact to produce a phenotype. So we can have these two different genes interact. So this is what is called two different genes interact to produce the phenotype. These two different genes, they are present on the homologous chromosomes. Sometimes the two different genes can be present on two different chromosomes. So it would be like this. So we have, these are homologous chromosomes. So we have D allele and D allele. So E allele and E allele. This is homologous chromosome. Same is the case we have here. X, small x, capital Y, small y. Sometimes these two alleles, this is one gene, and these, they will interact to produce the phenotype. So two different genes, this is one gene, this is another gene, they are located on two different chromosomes. They will interact to produce the phenotype of an individual. So two different genes located on different homologous chromosomes interact to produce the phenotype of an individual. This is also an intergenic interaction. So intergenic interaction, the genes can be present on the same homologous chromosome, or the genes which will interact in case of intergenic interaction, they will be present on different homologous chromosomes. This is the case of this is also a case of intergenic interaction. That means this is a non-allelic interaction. Then we have Batson factor hypothesis. What is Batson factor hypothesis? This is Batson factor hypothesis. Batson factor hypothesis that sometimes two or more than two genes influence the same phenotype or a particular phenotype in an individual. So we have two genes, two or more genes, or more than two genes, they influence the particular phenotype in an individual. So this is two or more genes. In some cases, two or more genes, they influence the same phenotype or particular phenotype in an individual. This is what is called as Batson factor hypothesis. This gene interaction and interaction between the genes, intergenic interaction, this is exception to interaction between the genes. It is exception to law of unit character. It is exception to law of unit character. So gene interaction is exception to law of unit character, as law of unit character says that one gene controls one character. But as we have seen in case of gene interaction, two or more than two genes can influence the same trait, same character, or same phenotype in an individual. This is what is an exception to law of unit character. Thanks.